I think this is where we left off. So let me review this slide and then we'll move on. So what had talked about, we talked about genetic mutations, and we'd been through a variety of different types of mutations. But we had gotten to looking at mutations a little bit differently. And if we don't mutate something, okay, so it's in its natural characteristics or has its natural characteristics, we consider that the wild type. And unfortunately, one of the labs that we'll be missing, we will take a wild type E. coli and we will transform it. We will add genes to it to make it different. And I wish we could do that, but that is too difficult of a lab to do from home. So a wild type would be the initial strain, and then a mutant strain would be the bacterium that exhibits the mutation. And in that case, the mutation isn't necessarily a bad mutation. It's something that we actually want to show that we can do for that particular lab, but it does tell us that it's different. It's different than the wild type. So in generic terms, there's two terms we use, and that's an auxotroph. And an auxotroph is something that requires a growth factor in order to grow. And that is, it means that the mutant cannot make or synthesize it, all right? And if we think back to amino acids and talking about amino acids, it would be a mutation, for example, when we want to make arginine, the amino acid arginine. It would be some sort of mutation in that operon and in that whole set of genes that works together. So there would be some sort of problem in that gene set, a mutation that's occurred, and now it is not able to make its own arginine anymore. And then there's the prototroph, and that's a bacterium that does not require a growth factor to grow. So in order to isolate mutants or mutations, organisms that have mutations, there's several methods to do that. But we have to think a little bit about what we're actually looking for. So if we have an antibiotic-resistant mutant, in other words, this is what our lab would have done. We would have put a plasmid in the wild-type E. coli, and if our plasmid was incorporated into that E. coli and was working in the E. coli, our organism, our wild type, would be sensitive to antibiotics or certain antibiotics, whereas our mutant would be resistant because it would have gained those resistance genes from the plasmid that we put in it. So we need to look at how we can tell things like that apart. Now, some things like fermentation indicator, that's going to be relatively easy, because if we grow an organism on a medium that allows us to tell the difference between an organism that can ferment versus an organism that can't ferment that particular sugar that we're looking at. So if you think about that, it would be maybe like our EMB, where our E. coli gave that green metallic sheen, but our Enterobacter gave more of a pink color. So that would give us an indicator right on the medium. When we're dealing with an antibiotic resistance, then what we can do is we can add that particular antibiotic to a medium. And if our organism is not able to grow, if it's the wild type, and then it doesn't have that mutation that allows it to be resistant, it can't grow. So a medium, in this example, a medium that has penicillin, and what'll happen is, well, let's compare first. So we've got a combination now of both penicillin resistant cells and penicillin sensitive cells. Our resistant cells are going to be our mutant. So we have a medium that has penicillin on the left-hand side, and we have a medium that doesn't have penicillin on the right-hand side. So on the right-hand side, we would expect all of our organisms to be able to grow, but only the organisms that have that resistance gene now are going to be able to grow on the medium with penicillin. So this example, and these are from your book, so when you're doing your reading, you'll see these, but let's say we want to induce mutations in organisms. And that's an example on this side, and we'll talk a little bit more about actually inducing mutations. But if we can induce mutations in certain bacteria, then those mutants, remember, in this case, this would be like us adding our resistance gene. So now we've got mutants here, and more of them would be able to grow on the medium with penicillin, and as well as the medium without penicillin. And remember that, and don't worry about that, but remember that the wild type would be what we start with. And that with the antibiotics, it's a relatively easy examination. Okay, it either grows or it doesn't grow, and the one that we're dealing with, usually we're talking about looking for an organism that has gained the ability in terms of it's gained the resistance to that antibiotic, so it will still grow. But what happens when we're talking about a mutation that shows a lack of something? So if an organism, I used arginine for example, if an organism can no longer make its own arginine, in order for that organism to grow, then arginine has to be incorporated into the medium. But the wild type will grow whether the organism is incorporated into the medium or not. So what ends up happening is we really can't see the lack of that growth. So this was a method that it's called replica plating, and this was a method that was used in order to see basically what doesn't grow, because that's what you have to look for. And I know that gets confusing, so, but I think if you follow this and you read your book, I think it'll become a little more clear. I don't have my board to write on while I'm doing this, so. But anyway, when we talk about, in this case, we've added a mutagen, and let's just say, oh, they use tryptophan, okay? So we'll use the amino acid tryptophan instead of arginine. But let's say that we have added something that causes or induces mutations. So what we need to picture now is our organism can normally make tryptophan. So it's got the genes, the genes produce the enzymes, and that organism can make its own tryptophan. But what happens now if we, instead of having it make tryptophan, we've now inhibited that gene. We've put a mutation in the gene, so now those enzymes are not made either correctly or in the right number or whatever, for whatever reason. So when we look at what happens when we plate it, if we inoculate on a medium, and when we say complete, it means it's got whatever is needed by the most sensitive organism. So in this case, it's a complete medium containing tryptophan. So it will grow anything, whether it can make its own tryptophan or not. So we put them all on here. Everything grows. But then we do what's called a stamp. And this stamp, this is a velvet surface. And you notice the X that you've got on the plates and the X that you've got on the stamp, and you line the two up, and that velvet surface, if you stamp that velvet surface onto these organisms, you're going to take some of each of those colonies and it's going to transfer onto the velvet surface. Then you imprint that on another set of media. One is going to be that same complete media that you had just used, but the other one's going to be a medium that lacks tryptophan. So if tryptophan is not present, then the organisms that can't make tryptophan anymore, those organisms that are mutated, are not going to be able to grow on this medium. So after 24 hours of incubation, and we look and we compare our media, and remember we've got these X's to kind of give you a location for each of these colonies, and we can see the colony that's missing. Okay, so we can see the difference. And this colony that's missing, obviously we can't take that colony and use it, but we could take it from here. And we know that that particular colony is mutated. It cannot grow. It is an auxotroph. It cannot grow without tryptophan now being supplied in the environment. There's another test that we use, and this has always been used to screen substances for mutagenic properties. So whether a substance can induce mutations. And I want to make very clear, this is only looking at an organism. And if you look at Salmonella typhimurium is what it used to be when this test was, that's what it was called when this test was established. It's now a Salmonella enterica serovar typhimurium. And what we start with is an organism that cannot synthesize histidine. Histidine is an amino acid, and it cannot synthesize its own histidine. That makes it an auxotroph. But what we've discovered is that certain certain substances that have mutagenic properties can actually back mutate. They can remutate that organism so that it goes back to the original, which seems a little odd. But if we think first about what we're starting with, our medium, in order to grow the Salmonella, our medium has to contain histidine.